Hello and once again welcome back to Excelsior Academy. So today we are going to do, as I said in the previous lecture, that we will be doing certain data interpretation type of question. So as a researcher, you should know once a given data is there, whether it's a histogram or some kind of graphs are there or the images are given. So you should know how to interpret the data so that you can understand what exactly is happening to your research experiments. Okay, so this is one of the examples which I have taken from the Cooper. So let's do this question. So this is based on the cyclin D1 and cell cycle progression. So I really appreciate that you guys should go actually learn all the cell cycle phases which I have already taken very nicely and in brief. So after that, come to these questions so that it becomes easy to understand what exactly being asked over here. Okay. So let's see what is the experiment first. The appearance of cyclin D1 during the cell cycle of normal human fibroblast was studied in this experiment. Okay, so it is normal human fibroblast, so around 24 hours incubation, that is the doubling time for the fibroblast, has been taken. Okay, now cultures were arrested in G0 by serum starvation and then stimulated by serum addition. Now this is very important to understand, because if you see the animal tissue culture techniques which have taken over there, serum is very important. Okay, because this provides the required growth factors, proteins, which vitamins which is required by the cells to grow. So that's why it is very important. Now bromouridine, a thymidine analog was added to the cultures at the same time. The cells were simultaneously stained with the anti-cyclin D1 and an anti-bromouridine antibody using immunofluorescence. Cyclin D1 and bromouridine specific fluorescence of 200 cells for each time point indicated in the diagram was determined. So here it is given. So you have got the time after serum addition, and this is the percentage of the cells. Okay, now the second, asynchronous cells and cells processed 24 hours after serum stimulations were stained with fluorescent DNA binding dye host and anti-cyclin D1 and anti-bromouridine antibody. Okay, so this was all about the experiment. Now if you see the graph over here, you can see that anti-cyclin D1 cells, initially they rise and then they fall, and then the other cells, that is anti bromouridine cells, the staining was done with the anti-bromouridine cells. You can see that there is an increase in the number of these cells. Okay, so let's see what are the questions. So what was the goal of bromouridine treatment? This is very simple actually, because already it is given in the experiment that it is a thymidine analog, right? So when it is incorporated in the cells, into that too in a newly synthesized, this bromouridine will be incorporated into the newly synthesized DNA molecules during the replication. So that's how you can actually track whether the cells are, dividing or not. Okay, so next question. What phases of the cell cycle are represented by the 10 and 24-hour time points in the figure A? So, okay, so they have mentioned over here. So these are the points they are asking. So in this case, during the period of 0 to 14 hours, if you see the percentage of cyclin D1 positive cells, okay, that is with the red line they have indicated, they increase gradually. So yes, with the time after serum addition, if you see, the cells are increasing gradually. So there is an increase in the cells. Now, what do we see that after a certain point, right? After a certain points, they decline and it goes less than even 10% by the end of the incubation period. So what does it mean? It means that these cells are in the G1 phases of the cell cycle. Okay. Now look at the other one. Now, at about 12 hours of incubation, that is over here, you see that the blue line starts increasing. You can see that there is an increase, right? So the number of bromouridine positive cells, they start to increase and they continue thereafter, even after the 24 hours. So you can see that there is a gradual increase and finally it keeps on increasing even after the 24 hours. So it means what? Since the number of the percentage is still keep on going increasing, it means that these are in the S phase of the cell cycle. So that's all. Okay, the third question, what is the relationship between cyclin D1 and bromouridine staining in figure B? I'm really sorry that I couldn't get the color print over here, but if you in future if in future if you get a certain example or certain graphs, then it becomes very clear. Okay, so you have got the host staining. So this is going to stain the entire DNA. So wherever DNA is present, it is going to stain the entire DNA. This is only for your cyclin D1 and this is only for your bromouridine. So the comparison is between the asynchronous synchronous cells and then 24 hours process cells after the serum stimulation. Okay. So what relationship is shown between the cyclin D1 and bromouridine staining is asked in the figure B. Okay. So what they're asking is, so as I said, as I mentioned before itself, this is your DNA binding dye. So it is going to stain all the cells. Then we have got the okay. So as I mentioned now, the cyclin D1 cells are in the G1 phase. So they are going to stain all the cells over there, the positive for the anti-cyclin D1 antibody staining. Similarly, anti-bromouridine antibody stains only the S phase cells. So what pattern, what relationship, what we see over here is that the staining pattern of cyclin D1 and bromouridine is opposite. It means that cyclin D1 positive cells are not positive for bromouridine and vice versa. That is, bromouridine positive cells will be there and then you don't have the cyclin D1 cells. So that means it is opposite. So that's what you see when you see the relationship between the staining pattern. So actually over here we don't have the colored images, otherwise it would have been very easy. So like this, the experiments are designed and you need to ask yourself like why I have used the bromouridine, why the staining was done. Now, what will be the positive control here, negative control over here? So that's why the host staining was done. So that whatever we determine, like in the confocal images, sometimes you might get, the false images to in order to confirm that those are the cells only. You need to do the host staining so that it confirms, yes, if these are colored, it means what these are the cells only. So you can see over here, these three cells, if you take, you see the what do you say? You see over here the imaging is there, but the opposite here the imagings are there. Like let me make it more clear for you guys. See, 1 2 these are colored among 1 2 and 3. So here 1 and 3 are colored, whereas here 2 and 3 are there. But you can see that two, even the third cells is not that much colored, right? Or the fluorescent has been shown. So that's why we said that the relationship is opposite. So where you see the staining with the cyclin D1, there the bromouridine staining was not there, and the vice versa. Okay, so similarly over here also you have got the 4 cells over here. The cells which are stained in the cyclin D1 are not stained with the bromouridine and the vice versa. So this is how you actually need to interpret the data. If any doubts are there, I'm happy to clear for you guys, or any other data if you have and not able to analyze your data. I'm happy to help you guys. You can mail me at excelsioracademy1228@gmail.com. So thank you for watching, and this is just for your benefit so that you can learn how to actually interpret the data. So thank you for listening. Keep watching, share and subscribe Excelsior Academy.